Good day, my dear postgraduate students. I would like you people to kindly admire the fish on the screen. Courtesy, this is the source from which I had taken it. A beautiful, multicolored fish. But the class today is on fish as such. I hope you people know what it is. Yes, it is fluorescent in situ hybridization, which is a university question for postgraduates. Guests like fish begin to smell after 3 days. Benjamin Franklin. His quotes used to be superb. So since the topic is about fish, I had taken a few of the quotes about a real fish. All men are equal before fish. Herbert Hoover. I didn't know who this gentleman was. I thought probably he was an author or a movie producer like the Moby Dick, Blue Whale. But then he happened to be the former president of the United States who handled the recession crisis extremely well. His quotes are also nice to have a look at. Even for small topics, think of the subheadings. The subheadings will take you to the answer. This is an extremely good disciplined action which I want you people to follow. So there is a definition, related terms. What is the basic principle? Types, applications, advantages, disadvantages. A diagram as such and any reference. So this is what we are going to deal with. My dear students, thank you for teaching me pathology. You are great. This is no flattery. It is not a business word or etc. It is the absolute truth. The students are the greatest teachers for the teachers. Look at this one. I am finding a couple of spirals and they are being interconnected by certain substances. This pale brown is the phosphate backbone of the DNA, and these are the connecting proteins. Adenine, thymine, cytosine, guanine. Just these 4 or 5 of them. See what havoc they play in designing your intelligence and my complexion. And also, look at this one. This is the final DNA that is going to be there. But sometimes when one is being lost, you find the new strand is able to replicate and rebuild the DNA. So this can be by continuous progressive action, which can come in the case of polymerase chain reaction. We shall see that in a different context. But today we shall be dealing with fish. See what is happening over here. It is a fluorescence in situ hybridization. So what happens is there is a probe DNA. Probe DNA is probably, you can take it to be the apparatus colloquially that is being used. And to a strand of the host DNA, you try to attach it. And in order to identify it, there has to be a label that is there. That is done by means of a fluorescent dye. So after denaturation and hybridization, I find that the experimental one gets attached to the host DNA which can be seen by means of fluorescence. And that is fluorescent in situ hybridization. It is a cytogenic technique developed by Christophe Lenauger. It vividly paints the chromosomes or parts of the chromosomes with fluorescent molecules. And it is used to detect the presence or the absence of specific DNA sequences in the chromosomes. After all, this is a chromosome and it is going to have the DNAs in it. In order to find out anything that is specifically present or absent, this can be used. Haptens. You must have seen it in all contexts, including immunology. You find that they are small molecules which can be combined to a larger protein so that they can elicit the production of antibodies. In fact, they will be helping in the antigen-antibody combinations. In this case, desoxygenin, fluorescent, spectrum orange, spectrum green. These we shall be remembered as haptens. And fluorophore is one that causes it to be fluorescent. So the elements that have been mentioned are fluorescent isothiocyanate, rhodamine, fluoroprobe dyes, oyster dyes, Alexa fluors, etc. A lot of these commercial names are there which can be mentioned. And the apparatus that is most important can be the fluorescent microscope. The fluorescence itself can be used for the RNA. RNA fluorescence can be used in situ hybridization, wherein FISH is a powerful tool to target any of this RNA transcripts, either in the tissue sections or in the whole mount preparations. And the FISH functions via the principles of nucleic acid hemodynamics, wherein two complementary strands of the nucleic acids. That is what I had mentioned in the first slide. They anneal to each other. Anneal means they become supplementary to each other. Therefore, they fix at exactly the precise points. Now, if there is going to be an error in this, then the whole exercise can be wrong. It can be from RNA to RNA or from DNA to DNA. Some books say as RNA and DNA complex. And this has been mentioned by Felsenfeld and Miles. So this is a hybrid. Now there is a broad selection options which are available to facilitate the tissue preparation, such as hybridization, post-hybridization, background removal, etc. These are all the steps. We shall see how it is being done. The annealing, as I said, is a combination. Strands of RNA and DNA can anneal to form the DNA-RNA hybrids. DNA or RNA probes are used to anneal to other DNA and RNA targets. So I can have DNA-DNA, DNA-RNA, etc. This is called in situ hybridization. That is mean in position. The earliest ISH protocols. ISH is simply in situ hybridization. This is the basic principle. Fluorescence is, after all, you are giving a color to the work. The radioactive probes were costly. Therefore, ISH was being used. And it required very long exposures. They were hazardous to health because of the radioactive probes. Methods such as fluorophores or probes became known as FISH. And these are the people, Rutkin and Stoller. Moreover, you can take computerized imaging of it for visualization and quantification. See what is happening over here, in situ hybridization. So this is a DNA probe, and this is probably the target RNA or the DNA. So here he says it is a mRNA and this is the target. So sequentially you find that they anneal to each other and they get fixed. But how do I know whether they are fixed or not? Then there has to be some kind of a hapten. So hapten is, see, it helps in the attachment of the antibody. It helps in antigen antibody reaction. Okay, the antibody is getting attached. How do I know it is present? There has to be a coloring substrate. It can be a chromophobe or it can be a fluorophobe or a coloring matrix, fluorophore. So this is what is happening. Simply you find that there is a probe and then this is getting attached to the target RNA and then there is the hapten, there is an antibody and then there is fluorescence. This simple diagram I think we can draw. So these are the components which you people must remember. Bye. And there are various steps such as the probe types, what are the published list of reagents, and the methods of detection. Look at this. This is basically a historical development of. Initially, as I mentioned, there was the radioactive ISH, Gall and Pardew, and Gall, 1969. Then came fish, Rutkin and Stoller, 1977. Chromogenic, it is not fluorescent, it is a color. ISH was used by Tanner et al. In 2000. RNA fish by Singer and Watt, and SM fish by Femino et al. And Raj et al. So look at the sequence of developments. If you go to the literature, you might find much more and better methods. So this is again the historical development. I had shown you a flow diagram of it. I will not be wasting my time again. So this is the technical development. I would like you people to kindly by heart the text because the previous diagram might be difficult for us to draw. This is easier to write. The fluorescent microscope. A fluorescent microscope works by combining. There is a magnifying property. After all, what is it? It is a microscope. Therefore, it magnifies the tissue. With fluorescence, there is the fluorescence that is being emitted. Look at this over here. You'll be having a series of filters, dichroic mirror, and there is an excitation filter. The object is there. Ultimately, on the stage, I get a fluorescence, and this is being spread. And it will not, you will not be seeing it with the eyes, but then you can do, but it is fed onto a computer where you get a permanent image of it. After all, any form of fluorescence, it has to be viewed within about 20 to 30 minutes, after which it will fade. Fluorescent microscope uses high intensity light source and that is called as a fluorophore. I would like you people to go to my classes on fluorescent microscopy. It is a university question and an essay question. What are the prerequisites? So the probes, I had mentioned it earlier. They are predetermined sequences which are used to get attached to the gene that is to be studied. In fish or ish, binding occurs only when parts of the chromosomes show high similarity. That is why the first picture of the DNA. So there has to be a similarity, matching, and a combination. Based on the size of the DNA fragments, they can be cloned with plasmid, phage, plasmid, and yeast. It is something like your immunohistochemistry. See, you cannot be continuously producing the markers with experimental animals. After all, the animal has to be sacrificed. So you need a continuous culture of it so that you get a specific antibody that is there. That is the basic principle for any immunological exercise. So the different probes that can be used can be from the centromere, central region of the chromosome, telomere, the ends. There can be the chromosomal bands, the G bands, and any particular location of the breakpoints. All these can be studied so that you know what is present, what is absent, or what is defective in that particular region. So this is the cloning of the DNA. See what is happening over. I'll try to explain it. So this is a collected genomic DNA and it is sheared. Sheared means virtually rubbed and then all the fragments are getting separated. The clone genomic DNA is being injected into the vector. Vector is the host and you find that it forms a combination with the DNA. And then it is getting transformed to the host strain, from which you can have initially a plate library. So almost like a culture plate, and you find things are growing. And then there can be a probe library for labeling it with a specific code. Finally, you find that the sequence identifying colony can be done. So this will be one of the things. And here again, it's a way of preparation of the code. So this is the one. Let us have an idea of it. But for us to remember, it will be better that. So I have this particular box you should memorize. Collect the DNA, share it, clone the genomic DNA, transform into the host strain, plate library, probe library, sequence identifying quality. I think this is simple. Now, what are the diseases diagnosed by fish? Fluorescent in situ hybridization. Identification of translocation breakpoints. Any of the chromosomal abnormalities you will be able to identify. Gene mapping. This is a newer technology where fish is being used. Deletions. If it is going to be gross deletion, a simple karyotype is adequate. But micro deletions, small points, can be identified by fish. Prenatal diagnosis to confirm inherited chromosomal diseases. Postnatal carriers of genetic disease. After delivery, whether they are still carrying it. Aberrant gene expression. So gene mapping, gene expression, I shall deal with it at a later date. Identification of new random abnormalities. And sometimes a 3D chromosome is also studied in the interface nuclei. Normally it is only a two-dimensional one, but here a 3D chromosome is also being studied at the interface. Regarding the infections, normal flora of the oral and the GAT, malarial parasites, and also to study the developmental biology, fish can be used. I think the entire list is worth memorizing. As I had mentioned earlier, there can be prenatal diagnosis of chromosomal abnormalities, as in the case of Edwards syndrome, Patau syndrome, Downs, Cry-du-Chat, chronic myeloid leukemia. Sometimes even in the diagnosis of ALL and AM. In CA breast, due to the extra copies of HER2. Monitoring the progression of a disease as well as the success of transplant treatment. Prenatal diagnosis of chromosomal abnormalities. It is a repetition over here. Infective organisms, flora, as well as the malarial parasite. Now, what are you seeing over here? So, P is supposed to be petite. Petite means short. And the long arm is labeled as Q because it comes next. So, there is nothing great about it. So, in this, they are finding the abnormality between this 5Q and 5P. So, this is being identified. So, it is a 5P syndrome or the Cry-du-Chat syndrome. And this incidentally is the banding technique. We use the GM for banding. So, the cell division, it is arrested at the case of, say, metaphase, wherein all the chromosomes can be seen very clearly. And then they are subject to GM stain wherein we will be finding alternate dark and light bands. Then we will be able to label them. Therefore, and you can send pictures of it all over the world. We will be able to find out where exactly things are abnormal. But this is a modern technique which is used, definitely an advantage over this. So this is from ResearchGate, fish for downs. So I find that there is a trisomy 21. So it's very clear over here. Trisomy 21 is over here, and with probes for 13 also, it can be analyzed. So all that I'm seeing is only the signal. I'm not finding the chromosome or anything. So the fluorescent probe is playing the trick over here. And the reference for this is PLOS Computational Biology. It is for the diagnosis of the malarial parasite. Normally, we keep searching for it in the RBCs. But then, look at these. You are able to diagnose it much easier. So this, incidentally, I would like you people to kindly go through the legend for the various diagnoses. The types of fish will be depending on the chromosomal cycle. It can be metaphase. Metaphase is always best because you find that the chromosomes get separated and then initially they will be somewhat tangled together. Then they get separated. Interphase will be a phase between the two divisions. They will be enhancing the fish technique. Sometimes a multicolor fish technique is being used. I'll show you a picture of it. One of the defects in fish used to be it cannot be quantitated. But nowadays there is quantitative fluorescent in situ hybridisation. And there is a nucleic acid that is used to quantify. A multicolored fish over here. But this is not what they mean. Here you find that the chromosomes are given different colors. So multiple probes can be attached, multiple fluorescents, and you get a multicolored fish technology. So this is a repetition of it. I would like you people to go through it. So there is a denaturation, the DNAs are being separated, and then there is a probe to which you are having the hapten and then the fluorophore getting attached, and after that you are able to identify. So there is an RNA fish to visualize the individual mRNA. This is called a single molecule RN fish, SM fish. Basically, they are all derivations of the ISH. So the steps are the same. I have mentioned it earlier. You people can go through and then write this. Metaphase. As I told you, the metaphase is used for this. Look at the chromosomes getting separated. Therefore, if at all you are able to find some kind of fluorescence, it will be quite easy. And this instance, BGR ABL in a case of CM. What are the limitations of fish? Sensitivity. When the RNA content is going to be low, the sensitivity is low. Cross reactions. It is something like false positivity or background staining. This has to be carefully dealt with. Little accessibility to the target site. Accurate hybridization and washing is required. Something like your ELISA, etc. There has to be an accuracy in doing the test. Quantification was initially difficult, but now it is being attempted at. Probably it is costly. All said and done, we are getting only the physical abnormalities. We are not getting any information about the physiology or the functioning. The nucleotides can act as quenchers and reduce the signals. Background noise. That is what we are going to see as fluorescent in the background. This is a problem with any of the fluorescent techniques. And the limitations can be expensive equipment, laborious, low resolution. It cannot be used as a screening method. Rather, it is used as a confirmatory method. You make a diagnosis of a leukemia and then you send it, you can get it confirmed. not the other way around. Structural abnormality within a single chromosome cannot be detected. There can be an error in the selection of the probe also. So this is again a repetition. It is for you people to patiently describe it. How it is being separated, how it is getting attached. The denaturation, it is done by means of this formamide at 42 degrees. This is a reference for it from Wikipedia. A wonderful one. Again, for a recapitulation, you find this is a probe DNA and it has got specific sequence. It has to anneal and get fixed to the host of it. So this is the host DNA. This is the probe DNA. They get fixed. And in a case of polymerase chain reaction, there can be never-ending sequences which can be produced, which is not there in this case. It is an in situ fluorescence that we are seeing. Fish can also be used for identification of the bacterial pathogen. The sample is collected from the patient, and then you are able to have that as a test sample, and then the fluorescent tag is being attached. And finally, you are able to treat it with any kind of chemical, etc. The fluorescence is finally expressed and the diagnosis is confirmed. Sometimes they say that there is a microfluidic chip wherein the cost has been lowered nearly 90%. This can be used. And there is a reference for it. I am not an expert at it. So this is just to show you at the metaphase, the human metaphase, which is being painted over here. And then we are able to study the fluorescence in multiple chromosomes. The reference is again over here. This slide is mainly to show you the advantage of the metaphase. And this is the centromere, and these are the arms of the chromosomes. This is one of the references. A lot of references I have already mentioned in the text. No good fish goes anywhere without a porpoise. What does it mean? It is not purpose. It is porpoise. Porpoise means it is a group. So as a group, we will be succeeding. So also the fish. Thank you.